Alright, y'all, this one's coming hot off the press. Yesterday, a group of scientists reported for the first time in human history, DNA editing has been used in vivo to save a human baby's life. Let's talk about it. There are two ways that DNA editing-based therapies can happen. One of them is called ex vivo, or outside of life. Basically, you take cells from a patient, fix the mutation that's making them sick, and then take those tweaked cells and put them back in the patient's body. This was the approach used in the world's first CRISPR-based therapy, Casgevy. Doctors take bone marrow cells out of patients with sickle cell anemia, tweak the mutation that's making them sick, and then put those bone marrow cells back in so that they can start cranking out healthy red blood cells. But there's another way to do this called in vivo, or inside a living body. Basically, you take the DNA editing machinery itself and inject it directly into a human patient, so all of the editing occurs inside of their body. And for the first time in human history, we've done it. A baby was born with a mutation that causes a disease called CPS1 deficiency. This is a lethal illness that basically turns protein into poison, but you still need to eat protein in order to survive. That's why CPS1 deficiency carries a 50% mortality rate in early infancy. Within just 48 hours, this baby was showing symptoms that were threatening his life. Doctors predicted that he would need a liver transplant before he was even 5 months old, but little tiny livers are very hard to come by, and the transplant process is extremely dangerous in a baby that's already so sick. So his parents did something incredibly brave. They allowed a team of scientists at Philadelphia Children's Hospital to try out a new in vivo DNA editing therapy to try and save their son's life. The scientists used a flavor of CRISPR called base editing. Basically, your DNA is made up of two strands that are connected in a double helix. Original flavor CRISPR breaks both of those strands, but base editing only has to break one of them, which means it's a lot gentler on your DNA. This amazing team of researchers, including one of the inventors of base editing, an evolution hacker who I made a whole video about not too long ago, injected DNA editing machinery directly into the sick infant. Within 7 weeks, doctors were able to increase the amount of protein that the baby was able to eat, halve the dosage of the medication that previously had been keeping him alive, and establish that he showed no adverse effects to receiving the treatment. Now, this is all very preliminary. This is only done in one patient and very recently, so there's a lot of work that still needs to be done, both assessing the long-term effects of the treatment on this one patient and increasing the number of patients that have received it. But this is a huge deal. For the first time in history, we have edited a person's DNA inside of their body, rather than in cells in a dish that later get transplanted back into them. And so far, it looks like it may have saved that human being's life.